neural communication today. We're going to have a look at how neurons work, what they are, how they work, what they do. Let's have a look and see what we can do here. I've got basically, I think I've zoomed in too much, there we go. I have got here an idea about why do we need communication. So we've got this idea of communication. Well, we've got basically a body, and this body needs to sort of communicate around and within itself, and we've got various things that we can do here. The first of these we can do is cell signaling, and cell signaling basically allows us to communicate with either neurons or chemically with hormones to send information around the body, so that we can basically transfer signals locally. And as well as that, we can have some sort of cell to cell communication. Plants don't have neurons, but instead they can use hormones to achieve some sort of communication from one to another. But as well as this, we've got then body to body communication, how one organism can talk to, communicate with another organism. So within the body we have these, and then we have some form of external communication, and this can even be with plants, that there is some evidence that plants can communicate from one plant to another. Not the sort of things that you expect, but basically some information about relevant attacks or something. So there are interesting sort of communication systems that we need to be aware of. Much of this signaling is all to do with homeostasis. Basically this idea of trying to keep the organism in a consistent and stable state. But we've also got the neurons, which will allow things like effective movement. So let's have a look at these, and we're going to start looking at the structure of a neuron. The neuron is probably one of the largest cells in the body, and basically the cell body of the neuron is sort of a little bit special. It's absolutely packed with large amounts of endoplasmic reticulum and mitochondria, because there we've got the production of chemicals called neurotransmitters. These are the chemicals used to pass signals from one neuron to another. So let's start with looking at a sensory neuron. So a sensory neuron is going to have a series of little, I suppose, connectors and these will then link into a central little tube, and this little tube somewhere along it will have the cell body, and this for going in is called a dendron. Going out is an axon, and this will basically end almost, I suppose, in the same way that the other one started, with these little tiny little connectors. So we've got these axons. Here's our cell body with our nucleus, and this is going to be packed, as I said, with endoplasmic reticulum and mitochondria, because we can't get mitochondria into these tubes. Surrounding these are special cells, these are Schwann cells, and they provide a myelin sheath for some of these neurons. Not all of them. Neurons come in different styles, and for control of things like muscles, then we will have these, but for other things we won't. So here is a Schwann cell, and we have this little gap between them, and this is the node of Ranvier. This basically provides here a myelin sheath, and this myelin sheath basically is a fatty layer that provides insulation to our nerve. So here is one type of neuron. Here is a sensory neuron. If we try, we can do a motor neuron, and a motor neuron has a slightly different sort of shaped sort of body here, and coming out of here we've got all our dendrons coming out here. Tend to be shorter than the other one, and as you can see, it's going to come out to multiple cells to connect. And then we've got here our axon. So these are our dendrites, because there are lots of them here. We've got our cell body, and just as before, we've got these Schwann cells, these cells providing a fatty layer, and again we've got our nodes of Ranvier, and more about these in a minute. So you can see it's fairly easy to make one of these, and then this is also going to terminate. Put another cell in there. So there is our picture, and what we see is communication going along this cell, and it only goes in one way. So our communication for these cells goes this way. And only that way. So this is really the types of neurons. We've got three. We've got the sensory neuron, we've got the motor neuron, and then we've also got a relay neuron. And a relay neuron basically looks much more of a mess, I suppose, basically. And it's going to have one sort of axon, and it's going to have a series of dendrites, so lots of those, and then we'll have just one axon. So this here, we've got information coming in via all of these and only going out on one of them. So in the same way, I could draw the same on this one, that we've got information coming in with these, but only going out on one of them. So the relay neuron is a sort of a much messier style, I suppose, here. And the way that we look at the communication here is simply that we start off with a receptor. This receptor then passes the information to the sensory neuron. The sensory neuron passes this to a relay neuron. The relay neuron may of course pass to many other relay neurons, but effectively we come out somewhere with a motor neuron, and that drives some sort of effector. So there is our basic structure of these neurons. These are what we call myelinated neurons, and these myelinated neurons covered with this myelin sheath made of many layers of plasma membrane. These special cells called Schwann cells produce these layers of membrane by growing round the axon many times. Each time they grow around the axon, a double layer of phospholipid bilayer is laid down. And when the Schwann cell stops growing, it could be sort of up to about 20 layers thick of membrane, and this acts as an insulating layer, and it allows these myelinated neurons to conduct electrical impulses much faster than the unmyelinated neurons. A myelinated neuron here can transmit perhaps up to about 100 meters per second. In comparison, a non-myelinated one would do perhaps just 1 meter per second. So these are used for communication when you want to move something fairly quickly, like a finger or an arm or a leg. And the non-myelinated neurons are used for controlling things that you don't need to do in so much of a hurry. And those sort of things are going to be, I don't know, move your gut or something. So we've got those sort of cells to do what we need to do for basic communication. Looking at then the Schwann cell, let's try and give you an idea of what this looks like. We've basically got our neuron going through, and this Schwann cell will put a double layer of phospholipid around there, and then we're going to have another one. There's our Schwann cell. The gap between, and you can see I've really well drawn this gap here, is the node of Ranvier. And these nodes here are a small gap about here, I suppose, 2 to 3 microns apart. They're not very big. The myelin sheath here is sort of, you know, quite long, so this gap here is probably in the region of 1 millimeter in sort of size. So what we have here in these myelinated neurons, the electrical impulse jumps from one node to another. So this is how this is going to work. So our electrical impulse is going to jump from there to there and so on to move along. This allows this information to transmit much faster. In a non-myelinated neuron, the signal doesn't jump from one node to another, and so the transmission is much slower. In some diseases, then some things basically go wrong. So you've probably heard of multiple sclerosis, and in multiple sclerosis, which affects some 100,000 people in the UK, and many of these people are diagnosed in their 20s, 30s, 40s, and this is affecting the nerves of the brain, the spinal cord, and it includes all sorts of problems with muscle movement, coordination, balance, and vision. This multiple sclerosis is an autoimmune disease where the immune system is mistaking your ordinary neurons for invaders, and they're being attacked, and there's a thinning or a complete removal of this myelin sheath, and this is going to slow down the signals and maybe stop them altogether. And the problem is here, we just don't know what causes this, and this is sort of a big problem. If we knew what caused it, then maybe we could stop it. But at the moment, for things like this, there are no cures. And not just multiple sclerosis, but we also see other diseases where the nerves can sort of almost die back. Things like peripheral neuropathy and others. So where does this all start? Well, it usually starts with something like the finger, and the finger being stabbed or feeling something with a pain, being sort of the finger being pricked. There are a whole load of sensors. People think there are only normally 5 senses, but in fact in your hand you've got that number of different types of sensors. You can detect things like pain, touch, pressure, heat. We can also detect things like water. Quite interesting that if you put your finger in water, you know it's water. But if you put your finger in a different liquid, it feels different. You can tell it's not water. So some of these nerves, quite clever what they can do. So let's have a look at some of these sensory receptors. So the sensory receptors, they are really very specific, and their job is basically to convert some sort of sense, I suppose, to an electrical impulse. So we can basically convert our taste, our touch, our smell, our temperature to an electrical impulse. Now, there are 4 different types of these. There are some mechanical types of receptors, and these work on things like pressure, movement, and an example of these are probably something like a Pernician corpuscle. Pernician corpuscle there, and we would find that something like in the skin. There are chemical receptors, and these obviously detect some sort of chemical. we would find these in perhaps something like the nose, in the olfactory receptors. There are the ones that can detect temperature, and these might obviously going to detect heat. These are the end bulbs of Carews. Which we'd find in the tongue. And we can detect light, and well, I suppose it is going to be light there as well here, and this would be something like a cone cell or rod cell, and we'd find these in the eye. So we can see we've got a whole different collection of these, and a name we would give these is to call it a transducer. A transducer is something that basically converts some sort of sense to an electrical impulse. So the sensory receptors can detect all sorts of different things, from touch, taste, temperature, light, sound, and they can generate an electrical potential. Let's have a look at one of those, and let's look at the Persician corpuscle. Now, what does this look like? Well, basically, we've got a nerve ending coming in, and when this nerve ending comes in, we've got our Schwann cells around here. Now, around this little nerve ending, what I've got is layers of connective tissue around this. There's certainly a lot of layers going on here. I'll draw a few more. And when we've got this, there's our basic sort of layers of connective tissue, and between them they've got, so this is connective tissue, and between all these is some sort of viscous gel. Around this we've then got some sort of blood capillary going around, so I might have a little capillary here going around, and around this we've got sort of some sort of other tissue, sort of epithelial tissue, forming sort of a layer around it. So here is our basic structure, and the important bit, I suppose, is to think of this little nerve cell. I'm going to highlight it in here so you can see which bit is the nerve cell. And there we've got our structure. Now this, as I said, is all to do with mechanical changes, and what we're going to see is this is what we're like when we're resting, but at a point when some pressure is applied, and if I just draw out a little bit of this, if pressure is applied, then these layers here are going to be squashed in. So let's just draw in the membrane. So it's at this point here that pressure is basically applied, and as this does so, then we're going to see some changes in here. So normally this is all negative, but at the point where the pressure is applied, then this changes to a positive. And if we look at here, normally outside here this is all positive, but where the pressure's applied, it's gone negative. So we've got here what we call a generator potential. This here, something has happened. So let's see if we can explain what's happening here, and we can get some ideas of really what's going on. Well, it's quite easy to sort of work out what goes on if we look at a series of instructions. So let's see if we can do this. Basically, we are at our normal state. And this we just call our resting state, or we'll call it our resting potential. So here everything is sort of okay. There are some sodium ion channels. These allow the movement, obviously, of sodium ions here, and they are basically normally set that they are now too narrow for movement. Now, pressure is applied. And what happens when the pressure is applied? Basically, we see a change in shape. We see a change in shape, and this is going to allow basically the membrane here to stretch. When this happens, the membrane stretches, and now we get the sodium channels, they widen, and that means that sodium ions, Na plus ions, diffuse into the neuron. The influx of these positive sodium ions here makes the membrane depolarized. And this creates something called a generator potential. This generator potential now creates our action potential. And this action potential is then transmitted along the neuron to our central nervous system. So, what is this action potential that I'm talking about? So, we need to have some idea about how a nerve works and what it does. And this is quite a long story, and so what I'll do is next time I'll go through how the action potential works and what it does. So thank you for watching this. Hopefully you've got a good idea of what a sensory neuron will look like, what a motor neuron will look like, what a relay neuron will look like, and you've got some idea about what some of these sensory receptors look like. I've just drawn one for you to give you an idea of that. The others aren't perhaps quite so obvious, like the chemical receptors. They don't look anything like the same. So next time we'll have a look at how an action potential works on A-level biology, topic by topic. Until then, stay safe and I'll see you later. Bye bye.